So an amino acid stretch of nonpolar hydrophobic amino acid residues preceded by positive charges from basic like arginine or lysine. But you can see that it isn't an exact amino acid sequence. So what does the ER signal sequence do? So what it is it's some nonpolar amino acids right here. And what it does is it binds to another protein or another particle that Günter Blogel's lab isolated called SRP, or signal recognition particle. Now originally, signal recognition particle was called signal recognition protein. And when they isolated it, they realized that it was more than just protein. There was actually a large, important RNA component, a structural RNA. So this shows SRP. This is a very large particle that's in the cytosol. And you can see the backbone of RNA right here that's holding the polypeptides together. So there are 6 or so protein subunits, one of which has GTPase activity. We're actually not going to talk about the GTPase activity, but it's important for its regulation. And it has this small structural RNA unit. So they called it signal recognition protein, realized they had RNA, and so changed the name to signal recognition particle. It worked really well. So what SRP does is it's a particle in the cytosol that will bind to the ER signal sequence. And it's in the cytosol. So to direct a protein into the ER, you need an ER signal sequence and SRP in the cytosol. And then you also need a channel for for the ribosome to secrete this protein through the ER. So recall that proteins can't cross membranes themselves. They need an aqueous pore or a channel. And that channel is called the translocon. Also isolated by the Global Lab. It serves as a channel for translating the protein, and it's on the ER membrane. So this shows the translocon. It's a super complicated structure that was actually crystallized, which was a feat in itself. It's really difficult to crystallize membrane, things that are in membranes. But the crystal structure was solved maybe 5 to 10 years ago. And it was found to be a multimeric protein structure that sort of looks like a channel. It looks like that, like a donut right here, with it's stuck in the membrane with an aqueous pore that the protein can go through. And the cool thing about this structure is it has 2 states. One is a closed state where it looks like there's a plug in there, preventing proteins or anything from going through the ER membrane. And then it can be induced to open up to allow proteins to travel through, right here. It's super conserved. Proteins similar to translocons this is in all eukaryotic cells, and structures similar to this are also found in prokaryotic cells. Okay, so how do we put these things together to look at how a protein is going to make it to the ER? And what we're going to use as an example is a protein that's going to end up being secreted. So this could be a protein like serum albumin that's going to end up on the outside of the cell. It could be a small peptide, it could be growth hormone. It could be insulin, anything that's going to be a protein that's going to be secreted to the outside of the cell actually begins life in the endoplasmic reticulum. And this is how it works. So we have messenger RNA for serum albumin or insulin or growth hormone. And there's nothing special about the RNA. There's nothing special about the ribosomes. At the 5' end of the RNA, the small subunit and the big subunit come together, and they start synthesizing the protein. And if there was nothing special about the protein, that protein would end up in the cytosol, as many proteins are. But remember that these proteins have an ER signal sequence at their amino terminus. So among the first amino acids that are synthesized are this ER signal sequence, and that's shown in this diagram in red. And what happens is SRP binds to that ER signal sequence. So here we see SRP, there's lots of these particles in the cytosol floating around, has a binding site for that ER signal sequence, and it binds tightly to that ER signal sequence. And once it binds to that ER signal sequence, translation stops. So the binding of SRP to that ER signal sequence stops the ribosome from translating anymore. And that hangs around until SRP complexed with the ER signal sequence binds to a receptor on the ER membrane called the SRP receptor. So we have a fourth ingredient right here, the SRP receptor. This will bind SRP ER signal sequence complex. And it will tether that to the ER membrane. Or where it is that's what it's going to do. It's on the ER membrane. The SRP receptor is an integral membrane protein on the ER membrane. So we've now managed to bring this whole complex to the rough endoplasmic reticulum, tethering it there. This ribosome is docked onto the ER membrane. And then what happens is this complex comes over to the translocon. That's shown here. And that the complex of the SRP receptor, SRP, and the ER signal sequence induces the translocon to open. And once it opens, once this aqueous channel opens, a hydrophobic patch inside the translocon is exposed. And the ER signal sequence has a tighter affinity for that hydrophobic patch inside of the open translocon than it does for SRP. And so it transfers, it will now bind the interior of that translocon. So once it is released, the SRP takes off, and it can go then to other messengers and other complexes, finding other ER signal sequences. And because it's released, translation begins again. The translation block is relieved. SRP signal receptor takes, the SRP receptor takes off too because SRP is gone, so they're both kicked out. And then the translation block is released, and translation will continue. The ribosome is docked onto this translocon, and so translation continues, and the protein ends up in the rough endoplasmic reticulum. So this is the story for proteins that are going to end up freely soluble on the outside of the cell. Proteins, again, like growth hormone, free proteins that are floating around in the extracellular matrix, begin life freely floating in the ER lumen, the inside of the ER. And they do so by using these ingredients in the order that I just talked about. Questions? Okay, so that's the process right here. So we have this protein being synthesized. We have another ingredient called signal peptidase right here. Signal peptidase is a protease that will cleave the ER signal sequence. It recognizes that sequence. It, in contrast to trypsin, is super specific. So the only polypeptide bond it's going to cleave is this one that's just downstream from the ER signal sequence. it probably recognizes something about those hydrophobic amino acids in combination with the positively charged amino acid. So it will cut that off, and once it's cut off, and the rest of the protein just falls through. So it'll cleave the ER signal sequence, and it's on the ER membrane. Now, looking at this diagram, you might think that the ER membrane is just chock full of ER signal sequences. Proteins are synthesizing a lot. And it turns out that this isolated ER signal sequence has never been isolated. It can't be purified because it's somehow, once it's in the ER membrane, it's almost instantly degraded somehow and gotten rid of in a way that we don't understand. So the way that Blobel actually found ER signal sequences was by applying drugs that would pause this whole process and then isolating the proteins and identifying ER signal sequences, as well as comparing the sequences of proteins that were going to be synthesized in the ER, going to end up on the outside of the cell, and realizing they were a little bit longer than the mature proteins that were found on the outside of the cell. Questions? Okay, so I just went through that step by step with those diagrams, but a movie is, a picture is worth 1,000 words, a movie is probably worth 1,000,000 words. And this movie is in the module folder as well. So let's look at it. This movie starts with showing the synthesis of a secreted protein with the process that we just saw. Movie leaves out some of the ingredients, so because otherwise things would get too crowded. But it shows you after protein synthesis has begun. So here's the message. The ribosome has already docked onto the 5' end of the message, the small subunit and the big subunit. And synthesis has already begun. We see the ER signal sequence emerging right here. SRP binds to that ER signal sequence at the amino terminus. Translation stops, pauses, and it will pause until the SRP plus ER signal sequence encounters the SRP receptor on the ER membrane. And then this 3-part also exposes to the translocon. The ER signal sequence transfers to the inside, SRP takes off. Translation can now continue because and this Pac-Man signal peptidase comes up and chomps off the ER signal sequence. This is showing everything pausing, but this would all be simultaneous, and the protein goes into the ER signal sequence, into the ER lumen. And you see the protein folding right there, and that's because there are chaperones in the ER called, that are analogous to HSP70. There are HSP70 homologs in the ER that are folding the protein while it's being synthesized. And this actually helps sort of pull the protein into the ER lumen as well. The protein's being folded as it's being synthesized. Okay, questions? Go ahead and look at that movie. I think it's super useful. Okay, let's go to a Instapoll question then. So, one. And no need to discuss because almost all of you got the correct answer. That protein, that mutant protein would be translated into the cytosol. So.
Object. I object. I'm just kidding. You might argue that it might get ubiquitinated because it's not proper.
That would also be a correct answer. So, but that's a subtle answer that I'm going to go through. Don't jump the gun.